Hello everyone. I welcome you all to Biology Lessons with Kalyani. Today we will be talking about another important topic, that is nitrogenous bases. Let's first see what are nitrogenous bases. Nitrogenous bases are a group of heterocyclic organic compounds. What is the meaning of heterocyclic here? Heterocyclic compounds are those which have other atoms such as nitrogen in their structure apart from carbon atom. These nitrogenous bases form one of the major components of nucleotide. We know nucleotides, what are they made up of? Nitrogenous base plus pentose sugar plus phosphate. Hence, nitrogenous bases are the building blocks of nucleotide. The sequence in which these bases are present specifies the genetic code and hence specifies the genetic information. If we see, there are 5 important nitrogenous bases, and on the basis of their structure, we can classify these nitrogenous bases into 2 types, purines and pyrimidines. If we look at the structure of purines, they are larger and essentially consist of 9-membered ring structure. This 9-membered ring structure further consists of one 6-ring structure which is structurally similar to pyrimidine and an imidazole ring structure. Both of this 6-ring structure and imidazole ring structure, they are fused to each other. Examples of purine are adenine and guanine. If we talk about pyrimidine, the structure of pyrimidines is smaller and they consist of a 6-membered ring. What are the examples of pyrimidine? Examples of pyrimidine can be cytosine, uracil and thymine. Often it is seen that we face difficulty in remembering purine and pyrimidine structure as well as the examples. Let us find a simple mnemonic for it which can help us in that. Now if you see purine, the word is small compared to pyrimidine, but the structure of purine is big. So remember the mnemonic like this: big purine got some apples and grapes and small pyrimidine cut it. So apples is nothing but adenine and grapes is nothing but guanine and cut is cytosine, uracil, thymine. Clear? Next is, how can we remember the nitrogen atoms? So remember this, they gave a new plate of fruits to all odd numbers except number 5. So new plate is nothing but nitrogen. So you put the nitrogen in all the odd numbers except number 5. So if you see purines, nitrogen atoms is present in number 1, 3, 7 and 9. And for pyrimidines, it is present in 1 and 3. Now, let us look at the counting conventions. For counting, if you see purines, then counting always starts from the top left nitrogen. The rule is that nitrogen always gets the first priority. So start from the top left nitrogen and you go in a counterclockwise or an anti-clockwise direction for the 6-membered ring. And if you look at the imidazole ring, you have to number it in a clockwise manner. So let's see. So we put number 1 for the top left nitrogen, 2, 3, 4, 5 and 6. For imidazole, this is clockwise, 7, 8 and 9. If we consider pyrimidines, then the counting convention is that we have to count from the bottom or the base nitrogen, and the counting is always in a clockwise direction. See, 1, 2, 3, 4, 5 and 6. I hope this is clear. Let's now see how we can make the structure of adenine and guanine. So start by making the base or the core structure, 6-membered ring and the imidazole ring. Just put the double bond in the imidazole ring, and where the both 6-membered and the imidazole ring is fused, do not put the other double bonds yet. We will put that after we put the side chains. Let's now see for adenine. If you see adenine, it will get one amino group at the 6th position. So remember adenine as 6 amino adenine. Okay. And if we see guanine, the amino group comes in number 2 position and 6th group gets the carbonyl group. So guanine is 26AC. A stands for amino, C stands for carbonyl. Easy to remember. Now we will see where this adenine and guanine or the purines attach themselves to the sugar molecule while forming a nucleotide. So the 9th position we have a nitrogen atom. N9 attaches itself to the first carbon of the pentose sugar, which is either ribose or deoxyribose, to form a beta glycosidic linkage. So N9 here is important. N9 is involved in the beta glycosidic bond formation. Clear? Now we have studied the structure of purines. Let's now have a look how we can make the structure of pyrimidine. So we remember we will start from the middle or the uracil. So make the structure of uracil first. Start by making the core structure. 6-membered ring first draw and just put one double bond. We will put the other double bonds later. Now if you see, a uracil has second and fourth position. At 2 positions it will have 2 carbonyl groups attached. So remember uracil as 24C or 24 carbonyl. Now look at the structure of thymine. It is exactly similar to that of uracil. What's the difference? So you have just an extra methyl group at position 5. So thymine is 5-methyl uracil. Remember uracil structure? Thymine is automatically done. Let's now have a look what is the structure for cytosine. Cytosine number 2 position, the carbonyl group remains intact as that of the uracil, but only 4th position gets an amino group. So remember it as 42AC. A stands for amino group and C stands for the carbonyl group. Clear? Here the glycosidic linkage is made with the N1. So N1 of the pyrimidines attaches itself to the first carbon atom of the sugar molecule or the pentose sugar during the nucleotide formation. Now that we have seen the structures of purines and pyrimidines, we will see some more important points about them. If we consider DNA, it will always contain ATGC and RNA will always contain AUGC. So, the purines if we see, that is adenine and guanine, it is common in both DNA and RNA. So, what is different? Pyrimidines differ in both the nucleic acid. DNA consists of thymine, whereas the DNA, it consists thymine. See, DNA contains thymine while RNA contains uracil. And cytosine is the common base which is present in both DNA and RNA. Let us now look at the base pairing of the nucleotide bases. Now, the nitrogenous bases are always complementary to each other. What does it mean? It means that in case of DNA, adenine will always form 2 hydrogen bonds with thymine, and for RNA, it will form 2 hydrogen bonds with uracil. So A always combines with T for DNA and A always combined with U for RNA. If we see guanine, then it makes 3 hydrogen bonds and combines with cytosine. So this complementary nature of nitrogenous bases is very important because it helps in the process of replication and transcription. It ensures that specific bases pair up and correct genetic information is passed on. So I hope you are clear about base pairing. By now you have definitely understood the structure of nitrogenous bases, how to make them, what is the counting convention, right? And how do we do the base pairing? You would have definitely got a very good idea about it. If you want to check nucleotides in detail, I am providing a link here with which you can click and check it out. Also, make sure that you hit the like button, share and subscribe. Thank you everyone. Have a very good day.